Hi guys, welcome back. We're going to start a kind of new series of videos, lectures, tutorials, whatever you want to call it. In this one we're going to be looking at exchange, right? Now we're going to discuss what that means, but essentially in F211 what that's going to include is things like the lungs, and the principles of what an exchange system needs. Okay, so let's begin. What is exchange all about? Let's look at a very simple cell. Okay, now actually make my life a little bit easier to make it a little bigger. You see that was almost a perfect circle. Not many people can do that. Now, right, so let's just look at this as a cell. It's got a membrane around it. Inside there's a cytoplasm, there's a nucleus, organelles. Okay. Maybe not so many organelles if it's bacterium. Now, any cell, now to understand the principles of exchange, we need to talk about things going into the cell and out of the cell. Right? Now, any cell, it needs a few essential things to be able to survive. Now, I'm thinking primarily of processes like cellular respiration. Okay? So, cellular respiration, for that, you need to have glucose going into the cell. All right? You need to have things like oxygen going into the cell. Yeah? To be able to carry out that process to produce ATP, and the ATP, the energy stored in the ATP, used to drive other metabolic pathways. Okay? So that's one thing to think about. Now you also need to think about, well, you know, if this cell is going to grow, and if this cell is going to produce its organelles, produce its enzymes, it needs to be able to build proteins. So if it wants to build proteins, it needs to get things like amino acids from the environment. If it wants to replicate its DNA, it needs to get things like nucleotides or otherwise nucleic acids in there. If it wants to build lipids, it needs to get fatty acids, glycerol, right? You know, any cell to maintain its watery environment, to maintain its cytoplasm, to maintain its balance of chemicals and ions, it needs water, yeah, and it also needs things like minerals, sodium, chlorine, sodium ions, chloride ions, things to, you know, make sure the water potential of the cell is right. Okay, so any living organism, it needs to be able to get these things from its environment. Okay, but on the other side, we also need to get rid of things. Okay, not get rid, that's very unscientific. You know, things need to be removed from the cell, or they, you know, need to exit, right? So here, let's just look at glucose and oxygen. What, you know, as a result of that, of the cellular respiration, we're going to be producing carbon dioxide. Okay, that we need to get rid of. We also need to get rid of things like other wastes from metabolic reactions. Okay, so other metabolic reactions going on, things like nitrogen containing compounds, they need to be removed from the cell, can't let them build up. So excess nitrogen containing compounds are also removed from the cell. Okay, so other, you know, you can call them waste products as a general thing. Okay, not to mention that, you know, other, well, let's not go anywhere else. Let's just think of the one cell for now. Okay, and you know, if this was a plant cell, for example, it would also be, you know, getting rid. I keep saying getting rid. Stop. It also needs to be able to, you know, remove oxygen from the cell. So oxygen also needs a way to need to leave the cell. Okay. I guess the main idea here, without, you know, having to go into the specifics, is that for any cell to remain alive, it's not just a bubble in itself. It needs to obtain things efficiently, and it needs to remove things from the cell efficiently as well. Okay, so for any cell to survive, it needs to be able to exchange. Okay, things need to move across the membrane of that cell. Okay, and that is called exchange. Some things need to leave the cell, some things need to come into the cell, and that's what exchange is all about. Okay. Okay. So one of the key features about exchange surfaces is the surface area to volume ratio. Now you've seen that. Hopefully, I'm going to explain that now, what that means, and it's going to make a bit more sense to you. So we looked at our single cell organism. All right. Now, you know, if we're thinking about a bacteria or a, I don't know, protoctist or something, or even a, you know, single fungal cell or a single animal cell, that one cell on its own doesn't have a lot of stuff inside. Okay, so a single cell on its own is pretty good at exchanging. Okay, because whatever it needs, it's got a very easy route into the cell. Okay, maybe I'm not making myself very clear right now. Okay, so if you've got some things coming in, some things going out, you can easily move across the membrane in and out of the cell. Now, so let's say we need oxygen and we are getting rid of carbon dioxide. Okay. Now for a single cell, that's pretty simple. Okay, but now let's think of a multicellular organism. Okay, now this is a multicellular organism, right? Now let's see about how easy it would be for all of these cells to be able to get the oxygen that they need and, you know, effectively release CO2 away so that the levels around it are not too high, so as to be harmful to the cell. So these cells on the surface, fine, great. For them, it's easy to get the oxygen that they need. But what about this cell, right? The diffusion distance is higher. And what about that cell here? Yeah, so from the surface to the center, yeah, the oxygen has to diffuse a great distance and through many barriers before it gets to that cell. Okay? So this is what we talk about when we say surface area to volume ratio. This cell has a very high surface area to volume ratio. Okay? Its entire surface area is effective and it is able to exchange materials. Okay, be that oxygen into the cell or carbon dioxide out. But if we look at this organism, all of its cells are not effectively able to exchange materials, only the ones on the surface. So this one, its surface area is low compared to the total volume of it, because there's a lot of stuff here that is not on the surface. Okay, and if we think of it in 3 dimensions, the problem is exaggerated. Okay, so this organism right here, it's bigger, it's multicellular, and it has a low surface area to volume ratio, okay, which means that it's not very easy for all the cells to get to be able to effectively exchange materials. If this cell produces carbon dioxide, it might leave the cell, but it's going to eventually build up in the area around it. If it builds up in the area around it, we're no longer going to have a very big diffusion gradient. So eventually the carbon dioxide will start to build up inside the cell, right? Causing a great deal of harm to the cell by, altering the pH of the cell, right? As well as that, it might not be getting sufficient oxygen, because oxygen has to diffuse through so many barriers over such a great distance. The amount of oxygen that it will be getting will be lower compared to the cells on the outside. Okay? And for that reason, multicellular organisms, need to develop specialized exchange surfaces, because they can't rely on diffusion alone to be able to provide all those things that we talked about before: oxygen, glucose. You can't rely on diffusion to take glucose to all your cells, oxygen to all your cells, remove carbon dioxide effectively from all your cells, yeah, because of this situation right here. Only a few cells here are able to effectively do that with the environment. Okay, so organisms that have a low surface area to volume ratio, and we are talking about the larger, any multicellular organism, it will need that. Okay. Okay, guys, right? So as organisms get larger, the need to develop specialized exchange tissues becomes greater, right? Because of this surface area to volume. Single-celled organisms or small organisms, they can rely on diffusion, because all of their cells are effectively close to the surface and can exchange directly by diffusion. But as your organisms get bigger, more and more cells, it's more and more inefficient to rely on diffusion to get to the cells within. All right, and in technical terms, it's all coming down to this surface area to volume ratio. This has a low surface area to volume ratio. This has a high surface area to volume ratio. All right, guys, we'll come back and discuss the specifics of exchange systems in a minute. In addition, you also, because you have more cells, you need more materials. So, I guess the key point would be that for organisms with a low surface area to volume ratio, because they are multicellular, it raises two things. First, there's greater diffusion distances, because the organism's getting bigger. Right? It's more distance from the surface where there's oxygen to, inside the organism where there might be cells also that need that oxygen. And secondly, so greater diffusion distance is one thing. The second thing is that there are more cells, so more materials are required. Okay? And so for this reason, you need specialized exchange tissues and organs, and cells even, to be able to A, account for the diffusion distance, and B, get enough materials to support such a large organism with so many cells. Alright guys, so when I come back we'll talk about what the features of a specialized exchange surface are. Hi guys, so I just wanted to spend a little bit of time explaining the surface area to volume ratio. It's not the most straightforward thing to understand. So what do we mean when we say high or low surface area to volume ratio? So this, let's just say that this is a small organism, this is a bigger one, and this is an even bigger one. Why do we say that this is a high surface area to volume ratio and this has a low surface area to volume ratio? Obviously this has a small surface area and this has a high surface area. So what we're talking about is the ratio of the surface area compared to volume. So this little cube here, for example, it has a surface area of 6 centimeters squared because it's got 6 sides, but its volume is 1 centimeter cubed. This cube right here has got a surface area of 54 centimeters squared. You can actually count the squares for yourself if you want to, but its volume is 27 centimeters cubed. Now if we divide, for this small organism, if we divide its surface area by its volume, we get a ratio of surface area to volume of 6 to 1. 6 divided by 1 equals 6. Here, for the larger organism, if we compare its surface area to volume here, we get 54 to 27, which is the same as 2 to 1. 2 divided by 1 is 2. So this organism has got a higher surface area to volume ratio of 6 compared to this organism, which has a surface area to volume ratio of 2. This organism has got a lower surface area in comparison to its volume. There's a lot of this organism which is inside, which doesn't get exposed to the outside. Alright? Whereas this organism, most of its surface is exposed to, all of its surface is exposed to the outside, but there's not a lot inside. There's not a lot of this organism that doesn't get exposed to the outside. Whereas this organism, there's a lot of it that doesn't get exposed to the outside. So it's got a low surface area to volume ratio, whereas this one has a high surface area to volume ratio. If you want, pause the video here and just compare the numbers for yourself and try and make sense of this. Alright? Alright guys, so we are now going to talk about what are the main features of a specialized exchange system. We've talked about why we need them. Now we're going to talk about what features they have and what features they all have in common. Right? Now, what I need you to appreciate here is we are talking about general things. The lungs, the digestive system, all and other examples are all exchange systems. They all show the features that I'm going to talk to you about now, and what you will have to do is to kind of remember these things when we talk more specifically about the lungs later on. Okay? So I'm going to talk about general things right here, and in the following videos, which you will find out about if you subscribe, and in the following videos I'm going to talk specifically about the lungs, but in this one it's more about what are the general principles that all exchange systems have, the lungs being one example of that. Okay, so let's talk about exchange systems. Exchange happens, things move from one area to another. See that? Again, perfect circle. Not, that's a skill. Not, so things move from one area to another because of diffusion. Now that, obviously it gets a bit more complicated. There's other ways that things can move: facilitated diffusion, osmosis, active transport, all those things. But let's just look at the, at the very basic level. Diffusion is what applies in most cases. So things move from one area to another down the concentration gradient. All right. So, for example, if you have a high, don't know why I drew that, if you have a high concentration of something in the cell compared to outside, there will be a net movement. Now these molecules are moving just randomly around, and these molecules are moving randomly around, but that random movement will eventually lead to, overall, some of these ending up out. Okay, and these guys will be moving around here. This one will be moving around here. This one will be moving around here. They're all moving around randomly, but I hope you can see that the random movement of ones in here leads to them getting outside. Okay, and that is what we call diffusion, right? Movement of particles, molecules, whatever you want, from regions of their high concentration to low concentration. And the steeper the gradient you have, the greater the difference between one area and the other, the more net movement of particles you will get. Okay? Right? So if I have this compartment, right, the random movements will make these concentrations of things equalize faster. There'll be a greater net movement the more difference there is in the two concentrations. So the lower that is and the higher that is, the more net movement I will get, the more net diffusion I will get. Okay? So it's very important that we understand that what we're talking about here is concentration gradient. Okay? If you keep those two concentrations different, you will get movement of one to the other. But as those molecules start to end up in this region, the movement of things from there to there will get gradually more slow until there will be no net movement in either direction. Things will still be going from there to here and from there to there, but overall the concentrations will be the same and there will be no movement. There will be no exchange in terms that are relevant to us right now. Okay? So exchange surfaces then, they have developed ways to maintain these concentration gradients. So first you've got a large surface area. Large surface area. So all exchange systems tend to have a large surface area. It might mean that cells might, fold their walls, their cell membranes in this way. right? So that cell has a much greater surface area compared to an equivalently sized cell or a similarly sized cell. Yeah, there's much more membrane here than there, much greater surface area. So this area is then well adapted to carry out any kind of exchange because it's got more surface over which to do that. Yeah, so most exchange systems have a large surface area. 2, they have a way to minimize the diffusion distance. So short diffusion distances are maintained by thin barriers. Notice I don't use any specific word like cell membrane, cell wall, cells, because a barrier can be many things, right? A barrier can be a whole cell. Yeah, for example, a capillary, for glucose to move, sorry, let's just keep on using the consistent example. For oxygen to move from the oxygenated blood out of the capillary and into the tissue fluid, it needs to cross the barrier. Now, in that case, the barrier is a whole cell. Yeah, it's the cells of the capillary. So a barrier can be a cell. If oxygen needs to move from the tissue fluid into the cell, in that case, the barrier is just the cell membrane. Okay, and if we're talking about plants, in that case, there's going to be a cell wall as well. So a barrier can be many things, and so to keep things applicable to many different circumstances, we're going to use the general term of barrier. Okay, so short diffusion distances are maintained by thin barriers. Okay. 3, coming back to our diffusion distance. Remember, diffusion gradient, a concentration gradient. We want to keep a high concentration close to a very low concentration, and therefore we'll get movement. So all exchange surfaces have ways to maximize the concentration of the material being exchanged on one side, for those 3, and on the other side, an efficient way to remove said material on the other side of the barrier. Okay? Efficient way to remove material on the other side. Alright? So if we're making sure that on one side we're maintaining a very high concentration, while on the other side we're making sure that we're quickly removing anything that does exchange or transfer or move, in that way we're keeping a very high concentration in one area and a very low concentration in another area, maintaining the diffusion gradient, maintaining the concentration gradient. Right, guys? So these 4 things are characteristic of all specialized exchange surfaces, and you pretty much apply this to many different examples, right? But these are the general principles. And next, when we talk about the lungs, I want you to be bearing this in mind, okay? Okay. I will now see when we talk about the lungs, right? Where I believe we will also be talking very specifically about how is it that the lungs have a large surface area, how is it that the lungs maintain short diffusion distances through thin barriers, and how is it that they maintain concentration gradients. Alright guys, so I'll see you then.